Hi. Hello, everyone. My name is Wacholder, and today I'd like to talk to you about statistical challenges in the detection of non-canonical proteins in mass spectrometry data. So first, what are non-canonical proteins? Non-canonical proteins are proteins that are not annotated in genome or protein databases, such as GenCode or UniProt. So if these proteins are not annotated, why do we expect that they might exist? Well, the main reason is a technology called RiboSeq. RiboSeq is recently developed technology which suggests that genomes encode many small proteins that are not currently annotated. Essentially, the way that RiboSeq works is that transcripts bound by ribosomes are sequenced, providing a genome-wide picture of translation, and this allows us to identify coding sequences that are not annotated. And the reason that these coding sequences are not annotated is because they are short, lowly expressed, and tend to be poorly conserved. However, despite the fact that these sequences are short, lowly expressed, and often poorly conserved, we know that some non-canonical proteins are biologically significant, and that's because individual studies have shown for particular non-canonical proteins that they do have important biological roles. For example, MLN is a 46 amino acid micropeptide which regulates muscle physiology in mammals, and since its discovery in 2015, there have been a steady stream of studies characterizing individual proteins that used to be non-canonical and showing that they are biologically important. At the same time, however, studies have also shown that many non-canonical proteins may be quickly degraded and likely have no biological role. So how do we distinguish these two classes? Out of thousands of non-canonical proteins that are potentially encoded by the genome, how do we know which ones might be biologically important? Well, one approach is to use proteomics. Proteomic detection of a non-canonical protein demonstrates that the protein exists stably in a cell and therefore could potentially have a biological role. And the main strategy used for proteomic detection of non-canonical proteins is called shotgun mass spectrometry. Shotgun mass spectrometry can validate that a RiboSeq predicted protein is actually present in the cell. The way that it works is that if you start with a database of RiboSeq predicted proteins, you can use an algorithm to predict the mass spectra that will be generated by those predicted proteins. Then you can conduct the mass spectrometry experiment to actually generate the spectra, and then you can compare the observed spectra to your predicted spectra, and if you get any match, it means that the protein is actually present in your sample. So this sets up the main problem I'd like to discuss with you today. RiboSeq experiments tell us that thousands of non-canonical sequences are translated. How many of their protein products can actually be found by shotgun mass spectrometry? Now, when we check the literature, what you see is that there's a wide variation among studies in the number of detected non-canonical proteins. Some studies find very few, while others find hundreds of them. In order to understand the factors that can explain why some studies find hundreds of non-canonical proteins and others find almost none, we attempted to replicate several of these studies, and today I'd like to talk to you about one particular replication we attempted in yeast. We attempted to replicate this study as closely as possible. So we looked at the same list of non-canonical proteins and the same list of spectra. We targeted the same 1% false discovery rate. But this original study identified 70 non-canonical proteins, while in our replication, we found none at all. So why did this original study find so many more non-canonical proteins than we did? In order to understand this, I first have to give a little bit of information on how confidence is actually obtained in mass spectrometry experiments. And the main strategy is called a target decoy strategy. Essentially, the way that works is that you have a target database, which includes all the proteins you expect might be present in your sample. And then from the target database, you construct what's called a decoy database, which is simply the reverse sequences of all your target proteins. And then you run an algorithm that goes through each of the proteins in either the target or decoy database and determines, based on how well these sequences match the spectra, whether the protein is actually present in the sample or not. And because we know that the decoys are not actually present in the sample, we know that anytime the algorithm says that it's detected, it's a false positive. And so we can use this strategy to estimate a false discovery rate as simply the number of decoys that pass the confidence threshold divided by the number of targets that pass a confidence threshold. So to give an example of what our results might look like, here I'm just going to plot confidence scores for all of the proteins in our analysis. And the confidence score I'm going to plot is called the expect score. what we need to know about the expect score is simply that smaller numbers indicate a stronger match. So here are expect scores for our decoys. You can consider this a null distribution. And here are expect scores for canonical proteins. You can see that many of them have very strong expect scores, much stronger than any decoy, indicating that we are very confident that these are genuinely present in the sample. But when we plot expect scores for non-canonical proteins, we find that they cluster together with the decoys. In fact, the expect scores of non-canonical proteins are statistically indistinguishable from that of decoys. And this is why we conclude that there are no genuine high confidence detections in this sample. Another way to look at this same data, we can look at a range of expect score thresholds and ask at each threshold how many decoys does the algorithm say are detected versus how many actual non-canonical proteins does the algorithm say are detected. Here are the decoys. And in order to obtain a 1% false discovery rate, we need the actual number of non-canonical proteins to be 100 times as large as the number of decoys as some confidence threshold. But when you actually plot the number of non-canonical proteins, it's very clearly nowhere close to 100 times as large as the number of decoys. In fact, it's a similar number. And so again, we infer there's no genuine high confidence detection in this sample. Why is it so hard to detect non-canonical proteins using mass spectrometry? In order to address this question, we looked at the factors that predict detectability among canonical proteins, and we find that two factors matter a lot. The first is basically the size of the protein, and the second is the expression level, here given in terms of ribosig reads. And so when we look at the canonical proteins, we find that it's very rare to detect them by mass spectrometry when they are below 50 amino acids long or below a certain expression threshold. And then when we look at the non-canonical proteins, what we find is that they're almost all below these detection thresholds. And so we predict that we should be able to detect very few non-canonical proteins, and indeed we detect very few, so there are no surprises here. Why then did the original study say that there were 70 detected non-canonical proteins? We think there are two main differences between the original study and our application that explain this difference. First of all, the original study used a proteome-wide false discovery rate, while our application used a non-canonical specific false discovery rate. So to explain what a non-canonical specific false discovery rate is, it simply means that we have a list of non-canonical proteins that we infer to be detected, and we ensure that no more than a target percentage of that list is false positives. On the other hand, what a proteome-wide false discovery rate means is that no more than a target percentage of the combined canonical and non-canonical lists, that is a list of the entire proteome, is allowed to be false. And what that means is that the way in which these false positives are distributed between the canonical list and the non-canonical list is not restrained in any way. And that means that if the false positives are distributed disproportionately among the non-canonical list, then the false discovery rate among the non-canonical proteins themselves can be very high, even as the false discovery rate at the proteome-wide level is quite strict. And indeed, we expect that this would usually be the case because, of course, there are lots of true positive canonical proteins that are very obvious. So we expect there to be a disproportionate number of false positives among the non-canonical list. And so using a proteome-wide false discovery rate rather than non-canonical specific false discovery rate, we expect will lead to a false discovery rate that's higher than the target false discovery rate. The second difference between the original study and our application that we think is relevant is that the original study merged protein lists from several distinct analyses. And to explain why this might be a problem, I'd like to go through a toy example. Suppose we look at two different conditions, and in each condition, we control a false discovery rate at a 20% false discovery rate. That means that if we detect 5 proteins, we expect 4 of them to be true positives and 1 to be a false positive. Now, what happens if we merge these lists in order to construct a total list of all proteins we believe to be detected? Well, because the true positives are genuinely present in the sample, they are real proteins, we expect that it's pretty likely that they are shared between both conditions. Cells express somewhat different proteomes between different conditions, but there's also a lot of overlap. But on the other hand, for the false positives, because they're not real proteins, there's no reason that they should be shared between conditions. And so when we unify these lists and look at the union, we expect that the false positives should accumulate quickly, while the true positives should accumulate much more slowly. So as you merge more and more conditions, your false discovery rate should go up and up. And so the result of merging multiple protein lists from different conditions is also that the non-canonical list can have a false discovery rate which is much higher than the target false discovery rate. And we think these two choices for how to analyze false discovery rate explains why this original study was able to claim 70 non-canonical detections, even though, as I showed, there is no difference in the confidence scores of the non-canonical proteins versus the decoys. So I wanted to point out that it is not our original idea that these are potential problems in mass spectrometry analyses. In fact, existing guidelines for mass spec interpretation warn against these issues, such as the Human Proteome Project Mass Spectrometry Data Interpretation Guidelines. Despite this, however, potentially misleading false discovery rate estimates are common in the literature. If we go back to the set of human studies that I showed earlier, we see that most of them actually use proteome-wide FDRs rather than non-canonical specific FDRs. In some of the studies finding very large numbers of non-canonical proteins also merge individually analyzed protein lists. And for these reasons, we think that the literature paints a misleading picture about the ease of detecting non-canonical proteins. So in conclusion, how many RiboSeq predicted non-canonical proteins can we actually find by shotgun mass spectrometry? We think very few can be found using current approaches. The low expression and small size of these proteins limit detectability, but we may need to use other techniques such as top-down proteomics or protein sequencing to find these proteins. More broadly, though, I think that this is a good example of a lesson that I think many of us computational biologists have had to learn a few times, which is that we really need to think very carefully about the statistical approaches that we use and whether they really capture the biological question that we're after. And with that, I'd like to thank you very much for listening and thank the members of the Carvunas Lab for giving a lot of helpful feedback on this project.
Quick question while the next speaker comes in.
Just really good talk. I think you communicated the science really, really clearly, and so that's helpful for non-mass spec people like me. I guess I'm curious how far off you think the technology is for small molecule discovery for applications like if you want to identify cancer neoantigens using these kinds of techniques and feel confident in the predictive ability.
Right, I'd say it's not very clear. There's strategies that might work, like trying to enrich for small proteins, doing very intense enrichment, or you might try other approaches like protein sequencing or more top-down proteomic approaches. I think it's really unclear right now what's the best approach for finding these small proteins.